So welcome everyone to the 40 somethings edition of this seminar series. It's my great pleasure to introduce Christine Jacobs Wagner today, and with her really intriguing title. I really very much look forward to your talk. Christine is originally from Belgium, and she was just complaining that the Belgian soccer national team was not doing so great. Um, but she has done her PhD in Belgium, then did a postdoc at Stanford, and had her lab at Yale since 2001. And in 2019, she moved to Stanford. Christine has received many awards. She's a member of the National Academy of Science and a HHMI investigator. And her lab has really been interested in bacterial physiology, biophysics of the cell interior. And really, the paper that introduced me to her work was this beautiful paper in 2014, where they saw that basically when you take energy away from cells, everything inside becomes glass. And that was really, for me, one of the initial papers that drew my attention to these kinds of problems. And so I'm really happy that you're here today, and I'm looking forward to your presentation.
Thank you so much, Gabrielle, for this really kind introduction, and also thank you for the invitation. Thank you, Yungpin, and thank you to all of you for coming for this talk. So I understand that for some of you it's very early or very late, so really I appreciate. And as you just saw, I had some technical issue. I lost internet connection. I hope that this is not going to happen again, but do let me know if that happens. I guess I will. I will probably notice myself. Okay, so I want to start with this movie. This is a very simple video, but I love it because it showcases the miracle of life. That is that the remarkable ability of cells to multiply. Now, what I think is amazing about this process is that for this to happen, there are things that are inherently random, such as the diffusion of micromolecules, that have to be transformed into spatial order, into cellular organization, and ultimately cellular function. And so I don't need to basically convince this audience that this is very important to understand how this happens, because that can tell us how cellular functions is achieved. It can also tell us about how cellular life is sustained, and perhaps it can even help us understand how cellular life began in the first place. So today I want to tell you about our recent efforts to try to understand to try to understand this question. And most of the work that I'm going to talk about is recent, and it was primarily led by this absolutely stellar senior scientist in my lab, Alex Papagiannakis. And I'm really thrilled to report that Alex is going to start his own lab in his home country, which is Greece. And so I think there's going to be a lot of exciting stuff coming out of there. Okay, so as Gabrielle mentioned, our model system are bacteria, and today I'm going to talk about E. coli only. And there's two reasons why we like to study bacteria. Is because one, understanding how they proliferate is important, since bacteria affects our health in more than one way. But also because because bacteria are simpler than eukaryotic cells. They're by no means simplistic, but they're certainly simpler. And so I think that we think that they are more closely resemble on how the ancestral cells might have looked like. Now, because this is a broad audience, I just want to remind you that there are big differences between bacteria and eukaryotic cells, starting by the fact that they're a lot smaller. I mean, this is just to show you the scale of how E. coli would look like relative to mammalian cells. Even if we compare it to microbial eukaryotic cells, bacteria are a lot smaller. Another thing that is characteristic of bacteria, if we look what's happening inside them, is that one, they're highly crowded. They're actually more crowded than the eukaryotic cells. And also they lack nuclear membranes, which means that the chromosome, which folds into a structure that we call the nucleoid, is going to be in the cytoplasm. And so cytoplasmic components can interact with the DNA, with the nucleoid. And we think that all those characteristics are very important, and they're important, for instance, for how things diffuse in the cytoplasm. And so to look at how things are diffusing, we use those nanoprobes that we developed back in 2014. This is actually the paper that Gabrielle was referring to, where we developed genetically encoded probe, which are made of a real virus protein fragments, mu and S, that we tag to GFP, and it assembles into a spherical particle that is diffraction limited. So that's really cool because then we can track it and we can see how things diffuse inside the cytoplasm. And if you express those in E. coli, they're going to form one particle. So that's convenient. And also a property that we really like is that they vary in size. So the size that they vary between is between 50 and 150 nanometer, so that we can see the effect of particle size with respect to diffusion. And so this is what Alex did. He tracked these particles, these nanoprobes inside E. coli cells that are also expressing a protein that is fluorescently labeled that binds to the DNA, so we can see what the DNA is. And it can get very long trajectory, as you see here. So this is just an example looking at 50 millisecond frame rate, but you also look at a lot faster. So with this kind of probe, so we can extract information about the diffusive characteristic properties of these objects that are diffusing in the cytoplasm. So we can measure the mean square distance that the particle move over time in the cytoplasm, and we get those kind of plots, so those MSD plots. And so by analyzing, for example, this is a log-log plot, if we look at the slope, this is going to tell us about the anomalous diffusion coefficient. And so if it's 1, then it means that the particle is diffusive. If it's less than 1, it is subdiffusive. And if it's more than 1, it's going to be super diffusive. And when Alex looked across the range of mu-ns particles, so between 50 and 150 nanometers, we see that that this anomalous diffusion exponent is close to 1. So it is diffusive. And it doesn't change much across the size of those particles. Now, this is very convenient because it means that we can extract the diffusion coefficient. And when Alex looked at the diffusion coefficient, the effective diffusion coefficient of these particles, so he's looking at thousands of them across the particle size, well, you see that larger particle diffuse obviously slower than that small particle. And that makes complete sense because things that are bigger are going to diffuse slower. But what does not make sense in the context of the Stokes-Einstein equation is that the relationship between the diffusion coefficient and the size of the particle should have a parallel of -1. And what Alex found is that the parallel is -3. And so if we look at this relationship and we plot what the viscosity should be, so the relationship should follow those lines. Okay, so in blue, if it was a low viscosity, and in red if it was a high viscosity. So it means that particles that are big perceive the cytoplasm much more viscous than the particles that are smaller. Even though it is exactly the same environment. And so that tells us that the cytoplasm is not homogeneous because, of course, the Stokes-Einstein equation assumes that the environment is homogeneous. And we know it's not homogeneous because, as I just mentioned, you have the chromosome, which is folded into the structure called a nucleoid that is inside the cytoplasm. And indeed, if we, this is what Jian and Ivan Suretsev, when they were in my lab, examined, is that if we look at the probability of finding any of these particles inside the cells, and you plot this probability, you see that the bigger they are, and the more they're going to be excluded from the nucleoid. And so this is what they plotted here. This is the probability of finding one of these particles anywhere in the cytoplasm. And if the particle is 50 nanometer, we also look at using a different nanoprobe at 20 nanometer, the probability of finding this particle is fairly homogeneous across the entire cells. But the bigger that the object becomes, the bigger is the nanoprobe, the more it becomes excluded. And now it's going to accumulate in DNA-free regions. And so this is convenient because now we can plot the exclusion score. So the more excluded it is, it increases with the size, as you can see. We can see that you start having this exclusion around 15 nanometer. And so from this, we estimate that the nucleoid mesh size is about 20 nanometer, 50 nanometer, I'm sorry. And so anything in the cytoplasm that is bigger than 50 nanometer, and this is consistent with what we've seen in the literature, are excluded from the nucleoid. And so as you can see, this anomalous diffusion is going to create some spatial order by affecting a large object inside the cells. And I come back to this in a minute. Now, I don't want to give you, there is actually another source of heterogeneity, is that when I say that the mesh size is 50 nanometers, or is estimated to be 50 nanometers, that is on average, because work from our lab and other labs have shown that the nucleoid mesh size is heterogeneous in itself. So on average, 50 nanometers, but then you have, and it's shown in the simulation here from Yinji, that you have region of the nucleoid where there is a lot of space and region where there is less space. So you have another elements of heterogeneity in the cytoplasm. Okay, so Alex reasoned that if we decrease this cytoplasmic heterogeneity, now the diffusion should be less anomalous. And so to decrease this cytoplasmic heterogeneity by diluting the DNA. So what he did is that he used a mutant, a temperature-sensitive mutants. When you switch to the restrictive temperature, the cell is going to stop replicating DNA. But it's still going to elongate, so it continues to grow. So effectively, you are diluting the concentration of the DNA. Plus, my lab and Kees Dekker's lab have shown that in these cells, the nucleoid is going to decondense. So it's going to get wider, which means that the mesh size is going to increase. And Alex shows that in these DNA replication arrested cells, now he's going to track those nanoprobes. And now he found that the power law becomes -1. So the diffusion coefficient as a function of the size of the particles now follow the slope is -1, which is, so that approximates what we, it is consistent with the Stokes-Einstein equation. So this is consistent with the idea that the nucleus mesh size contribute to the anomalous diffusion that we observe. Okay, so clearly the presence of the nucleoid in the cytoplasm affect cytoplasmic dynamics, especially for large cytoplasmic components. But it helps us understand why some cytoplasmic components have a non-homogeneous distribution inside the cells, and that helps organize the cells. But these observations also help us understand an old mystery. And that old mystery is what drives nucleoid segregation in E. coli. And before I talk to you about this, let me give you a little bit of background. So E. coli, just like any other cells, must replicate and segregate its DNA for cellular replication to be faithful. So in the context of bacteria, so you have the nucleoid, DNA replication is going to occur, the size of the nucleoid is going to expand. And then when the nucleoid reaches a certain length, that's what we've shown back in 2024 by Sandra Govers in my lab, the nucleoid is going to split, okay? So it's going to split, and now you have two objects, and those two, so you first have the nucleoid splitting, and then the two nucleoids are going to move apart from each other. So, and those two processes, splitting and the migration, I'm going to refer to them as nucleoid segregation. Okay, so you can look at it in this way. So if you project, like if you look at the nucleoid signal, if you, for example, if you stain it with DAPI, or if you use the fluorescent, if you use a protein, a DNA binding protein that's fluorescently tagged, so you can look at the nucleoid signals. And if you look along the cell lengths, and then you plot it over time or over the length of the cell, so from birth to division, you see this is a nucleoid, this is a relative cell length, so the cells normally gets bigger, but of course here we normalize for cell length. You see that the nucleoid segregation is going to occur at a specific time. So another long-standing mystery in the field is that not only what drives nucleoid segregation in E. coli, but also what control this timing. And in fact, this is one of the few things where we understand we understand better what happens in eukaryotic cells than it happens in bacteria. And so we know on beautiful work from the field of eukaryotic cell biology and genetics and biochemistry that eukaryotic cells use this beautiful mitotic spindle, this mitotic apparatus for segregating duplicated chromosomes. So you have a dedicated machinery that is very complex, involve a lot of proteins. But what about in E. coli? Well, as far as we can tell, decades of works have led to the discovery of no dedicated system. So there was no apparent cytoskeletal system involved. Now, if you do a search for bacterial chromosome segregation, you'll find a lot of paper, and most of the paper is going to be about the PAR-ABS system. Now, I love this system. My lab has worked on the PAR-ABS system. It's found in many bacteria, and it is important for segregating the duplicated origin of replication. So most bacteria have one chromosome. Each chromosome, the chromosome has one origin replication, and as soon as when you have replication, you have duplication of this region, and then the PAR-ABS system is going to be important for segregating this region apart. However, a little dirty secret in the field is that if you remove this PAR-ABS system by genetic deletion, well, nuclear segregation is still fine in most bacteria. And case in point, some bacteria don't even have the PAR-ABS system, and E. coli is a prime example. So it doesn't have a PAR-ABS system, and yet nuclear segregation is just as faithful in bacteria than it is in bacteria that have the PAR-ABS system. So we know that we are lacking something. Now, there has been really beautiful work that has proposed other mechanisms, and I'm not going to go into details. I'm happy to discuss it during the Q&A, so such as DNA replication, DNA loop extrusion that is driven by SMC or condensin-like protein, cell growth, translation, and so forth. And I do believe that they do play a role, but we know that any of those mechanisms can contribute to the splitting and the migration to the full process. So we knew that something was missing. And I'm going to kill the suspense. I'm going to tell you right away what we think is important, what is contributing to the nuclear segregation, and then I'll show you some supporting data. And so we think that the reason the work from Alex and the experimental work from Alex and the modeling work from our wonderful collaborator, Shiwei Hu and that Green at Princeton suggests that the reason why it has been so hard to identify a dedicated system that is involved in nuclear segregation is because it doesn't exist. Instead, what we think happens is that it is going to be an emergent property. This is going to be an emergent activity that arises from the existing activity. And that existing activity is, of course, we go back to the central dogma of molecular biology, the flow of genetic information. So if there is something that the DNA is very important for, it's for gene expression. And we think that the fact that it's involved for gene expression is going to contribute to chromosome segregation. So DNA is going to be converted into RNA by RNA polymerase. And then the RNA, the mRNA are going to be translated into proteins by ribosome, and that's going to result in growth. Now, in the process of, in this process, there's going to be the production of polysomes. So polysomes are mRNA that are loaded with ribosome. So I'm just going to use the term polysome to consider mRNA that have one or more ribosomes. And so ribosome, this is important, ribosome are about 20 to 23 nanometers. So the size is small enough to go to cross the nucleoid and to load on the mRNA that are made in the nucleoid, because this is where the DNA is. However, polysomes are bigger, about the size of 50 nanometer or are bigger. So they're going to be sterically, it's going to result in a steric repulsion from the DNA. But they are born in the nucleoid because this is where the ribosome are going to be loaded. And so the work of Alex and Shiwei and Matt shows that by considering the synthesis and degradation of polysome plus steric repulsion, this is going to be enough to contribute to nuclear segregation. And so what I'm showing you in this movie here is that here this is the DNA and here is the polysome. So to visualize a polysome, we label a ribosomal protein to a fluorescent protein. And since most ribosomes are in polysome form under those growth conditions, we are mostly looking at the localization of polysomes. And as you can see, it appears at the right place at the right time. Okay, so the way that Alex have been got into this is by analyzing the dynamics of polysome and nucleoid in those in this mother machine type microfluidics, where the bacteria are growing under very balanced growth in these little channels. And so we're looking at hundreds of channels, and it's balanced growth. So we can look at hundreds of thousands of division events. So this is looking at the ribosomes, and this is looking at the DNA. And by looking very carefully at the dynamics of those two micromolecules, he observed that there was a beautiful correlation between the two. So this is a chymograph from birth to division. And at this growth rate, the timing of nucleotide segregation occurs early during the division cycles. But as you can see, as you can appreciate, I hope, is that you see that not only the polysome accumulate in DNA-free region that is at the end of the cells, but also you have a particularly prominent accumulation in the middle of the nucleoid, and then they keep accumulating as the nucleoid is separating. And we can see here that the accumulation of polysome in the middle of nucleoid correlates at the time of nucleoid splitting. If anything, it initiates a little bit before, so it precedes it. So it's not only that the polysome are there at the right place at the right time, it's also that the rate of polysome accumulation correlates beautifully with the rate of nucleoid separation, of nucleoid splitting and nucleoid migration. And so this is illustrated here. So each data point here shows you the rate of polysome accumulation across cells at any given time. And you see that you have a beautiful correlation. And what I like most about these data is that the linear fit ends at the 0,0 intercept. So that means that in cells for which polysome accumulation is 0, the rate of nucleoid splitting is also going to be 0. So then let's break it. So let's break the mechanism. Let's stop polysome production. We can do this by adding rifampicin. So Alex, add rifampicin, which blocks transcription. And so if you block transcription, you're going to block the rate of polysome accumulation. So that's the first effect you're going to get. And then, of course, we can also remove rifampicin, and then you're going to restart. So we can look at the dynamics. So there is two effects from rifampicin addition. One, you block transcription. So you stop the rate of accumulation of polysome. And second, because you have mRNA degradation, it means that the polysome you're going to deplete polysomes because of mRNA degradation, and that occurs in the minute time scales in bacteria. So you're going to have two effects, a fast and a slow. One, stop the synthesis of polysome, and two, deplete the existing polysome, and that's going to be a slower effect. Okay, so I'm trying to change the slide. So we can do this kind of analysis. So what Alex have shown is that before rifampicin is added, well, nucleoid segregation occurs normally. You see that this is looking at the separation between two duplicated nucleoid from the center mass, and you see that like they move apart from each other. Now, if you look at cells that are born just before the rifampicin addition, first they segregate the nucleoid just normally. So the blue dotted line shows you the curve of this curve, and you can see that they match perfectly. But as soon as the rifampicin is going to hit the cells, as soon as it's going to be exposed, transcription is going to be inhibited, and that stops nucleoid segregation. And as you have depletion of polysome, you're going to reverse nucleoid segregation. And this is probably my favorite experiments. And so the reasoning is that the reason why this works presumably is because polysomes are going to be formed in the nucleoids, and they're going to accumulate there and then eventually split the nucleoid after duplication of this nucleoid. But what if we now recruit the formation of polysomes somewhere else inside the cell? So what if we have an ectopic polysome accumulation? And to achieve this, what Alex did is that he introduced multi-copy plasmid that express a useless protein. This is just a fluorescent protein, but under a very, very strong promoter. And so now transcription is going to be recruited at this location. And so it's going to steal away some of the RNA polymerase from the chromosome, and the ribosome are going to be attracted to this location. And this is where polysomal accumulation can occur. And this is an example of a cells. Not only polysomal occurs in the middle, which is usually what happens, but also it starts accumulating at the pole of the cells to a very large level. Presumably it's because you have the plasmid accumulating there. And we know it makes sense because the plasmid are bigger than the nucleoid mesh. And what happens is that the nucleoid segregation stops. If anything, because you have this large accumulation of polysome, it effectively push away the nucleus so that now they fuse together and they are nicely sterically repelled there is a nice steric repulsion between the nucleoid and the polysome, which are ectopically located. And we can see this correlation, this correlated dynamics across cells. And here I'm just showing you another example. This is a chymograph. Now, often when we overexpress this useless protein, the cells filament, so they get really long, which is what happened here. But this is kind of cool because we can see that because the plasmid move around, that the ectopic localization of polysome can change its of place. And you can see how this affects the nucleoid dynamics. And by the way, nucleoid replication, so DNA replication still proceeds in these cells. So as you see in the beginning, you have two nucleoids that are separated, and you have the accumulation of a polysome. And then you have the plasmid that accumulates at this end, which is where you have a huge accumulation of polysome, and that push away the nucleoid. Now they fuse with each other. And then a little bit later, clearly, now we have an ectopic accumulation of polysome at the other end of the cells, and that pushes the nucleoids in the opposite direction. And the reason also I show this particular chymograph is because you see this little gap here. This is actually a protein aggregate. Because we're making so much of that useless protein, it accumulates into inclusion bodies. And the polysome themselves are able to push this protein aggregates. And I think that this illustrates how crowded the cells are. Okay, so then the question is that, well, do we have all the ingredients? Is this enough to achieve this nuclear segregation? So as we understand it, we have mRNA expression happening in the nucleoid because this is what the DNA is. The ribosomal subunit are small enough, as we know, they diffuse through the nucleoid, and this has been shown by Yoanov and others. Now, because of that, it means that they're going to load on the mRNA and the polysome are going to form within the nucleoid, which we know it to be true. Now, the polysome are in the nucleoid, they're going to diffuse, but they're going to diffuse slowly. And we know the diffusion coefficient, which has been measured by our lab and many other labs, and we know it to be slow. And then, of course, you're going to have this steric repulsion between the polysome and the DNA. Plus, you have mRNA degradation, which means that there is a certain rate of polysome degradation. And the question is that, is this sufficient to obtain nuclear segregation during growth? And to look at this, we collaborated with Ned Wingreen and his wonderful student, Shi Wei-Hu at Princeton. And so what they did is that they developed a minimal reaction diffusion model. And it's this model. It's a 1D model. You have a repulsion between polysome and DNA, which is achieved through the steric exclusion process, just because the polysomes are bigger than the average mesh size. They assume that polysome production occurs uniformly within the nucleoid. Okay, so we are not, in the model, we don't have the polysome production happening in the middle of the nucleoid. They happen uniformly across the nucleus, so we consider the trivial case. We know what is the how fast the polysome diffuse, so they put the experimentally determined diffusion coefficient. We know the rate of polysome degradation, which you assume to be uniform. And we know that during growth, the nucleoid and cell size is going to scale. We've shown this in the previous paper. Okay, so they start the simulation, and at steady state, when the cells is small, what they see is that the polysome in blue are going to be excluded from the nucleoid in red. What is really cool is that when you add growth, and when you add growth, of course, the cell is going to become bigger. And what happens is that you have accumulation of polysome inside the cells, but inside the nucleoid, but particularly in the middle of the nucleoid. This is going, when it reaches a certain threshold, it's going to result in a phase separation. And through its continued accumulation, is going to push, not only split the nucleoid, but also push them away. And so this shows that just having these ingredients is sufficient in the model to show this splitting and nucleoid segregation. And so the way it works is because you have out of equilibrium processes where you have the synthesis of the polysome that occurs primarily, that occurs in the nucleoid, which means that it is higher than the polysome degradation in this location, whereas the rate of polysome synthesis is lower elsewhere relative to the degradation of the polysome. Now, the reason why we have an accumulation of the polysome specifically in, I'm sorry, I have a little problem with my laser pointer, like in the middle, is because this is where you have the lowest probability of polysome escape. So let me show you this schematically. The idea is that the polysome that are born in the middle of the nucleoid are going to have a lower probability of escaping the nucleoid than those that are in the middle. And so this is going to result in a gradient and an accumulation of polysome in the middle of the nucleoid. And what we observe is that this occurs when the nucleoid reaches a certain length. And this is consistent with previous work from our lab that shows that nucleoid segregation initiated at a certain nucleoid length across thousands of different genetic and metabolic perturbation. Okay, so as probably some of you are thinking now, it seems like the cell width of the cells might be important to achieve this localization pattern. So now what we see inside the cell is that the nucleoid here is close to the membrane, to the cytoplasmic membrane. It's for two reasons. One, you have this radial confinement, but second, we also know that it's going to be, the nucleoid is going to be attached to the cytoplasmic membrane by virtue of this coordinated transcription and translation and insertion of transmembrane protein. And so it means that the nucleoid is forced to elongate along the cell length, so that you're going to have the splitting specifically along this dimension, such that the polysome that accumulate in the DNA-free region and in between the nucleoids cannot have a tough time diffusing and fusing with each other. So this accumulation have a tough time diffusing with each other. And consistent with this idea is that if we increase the cell width of the cells, and we can do this by affecting the way that the cells grow, so using some drugs, if the width of the cells become a lot bigger, I mean, I'm talking about 5 to 10 times bigger, now we're going to have this phase separation in which now all the polysomes are going to get together in the middle of the cells, and the DNA is going to be all around the cells. And so this suggests that there is the radial confinement, so that the control of the cell width is important for this proposed mechanism to work. And this, in fact, reminds me of really a beautiful observation that Jeff Erickton's lab made a few years ago. So they work on bacillocellulose L forms, and those L forms are cell wall less. So they remove the cell wall of those cells, and those cells, under certain conditions, are still able to proliferate. However, they basically have many different sizes. They are spherical. They form spheres of different sizes, and the proliferation rate, they can proliferate, but it's very sloppy. And one process that is particularly sloppy is nucleoid segregation, which is shown here. So that this big cells is going to form a lot of little cells. Some of them are going to get DNA, and some of them are not going to get DNA. So this is, and people have proposed that this is maybe how the primordial cells would look like. Okay, so that we would have inefficient proliferation due to inefficient DNA segregation. But what is really cool, and what they observe, is that if they put those wall-less L-form cells and they force them to grow in those very narrow channel, now chromosome segregation is fine again. And that is because we think it provides this radial confinement that prevent polysome accumulation at a different location inside the cell, so in different DNA-free region, to fuse with each other, and which of course would affect nucleotide segregation. Okay, so then the next question is that if this drives nucleotide segregation, but what regulates the timing? So what we know is that the timing of nucleotide segregation is going to change during if you change the growth rate of the cells. So you can do this by affecting the you can do this by changing the growth condition. So in cells in which the growth condition is such that there is a slower growth rate because the nutrient quality is lesser, is not as good, then nucleotide segregation is going to occur late during the division cycle, whereas it occurs early if the cells grow fast. So is there a dedicated mechanism to ensure this, to control this timing? Well, the beauty of our proposed mechanism is that it inherently coordinates nucleus segregation to growth rate. Because imagine a scenario where now the RNA and protein synthesis goes down. So the growth rate, that is going to reduce the growth rate because you make less protein. But because you also have a slower rate of polysome production, that is also going to slow down on nucleus segregation. And the timing is going to be later. Whereas if you do the opposite, now you grow it under the rich condition, RNA and protein synthesis is going to increase, you have a higher production of polysomes, and you're going to increase the biomass growth, but you're also going to increase the rate of nucleotide segregation as well as its timing. And so what I love about this proposed mechanism is that the regulation is built in, right? Where you don't need a checkpoint, you don't need to evolve a dedicated machinery or a dedicated molecule, regulatory molecule. Instead, it occurs intrinsically. And that's what we observe experimentally. So if we look across 30 different growth conditions by varying the nutrient conditions, if the cells grow slowly, the nucleotide segregation is going to occur late during the division cycle. So this is between birth and division. But the production of polysome is also going to occur late. And if it happens early, the nucleotide segregation occurs early because you have a fast growth rate. You also have this correlates with an early and fast and higher rate of polysome production. And we look, this is just showing you 6 different examples here, but we look across 30 different conditions and we have a beautiful correlation. And this is also observed in the model. If now we change the growth rate, we know that the rate of polysome production changed. By just modulating the rate of polysome production, you can modulate the timing and the speed of nucleotide segregation. So this would be at fast growth rate, this would be a slow growth rate. And if we analyze, the data from Ned Ringgreen's lab with net experimental data, they agrees very well. And so what we propose here is that spatial organization, that the intracellular organization and the dynamics and the regulation that we observe in cells can emerge from basic cellular activities, and as long as you have constrained diffusion. And we think this is exemplified by the transfer of genetic information that contribute to chromosome segregation. And not only it drives chromosome segregation, we think it also intrinsically couple it to the growth rate. And so that goes back to the fact that gene expression occurs at the DNA level. And so if you're interested in that kind of work, I also encourage you to see a beautiful paper from the Mannix lab that touch on many of these points and some others. And the reason why I'm really excited about it is because emergent cell cycle activity and intrinsic coupling with growth rate is particularly attractive when we think from a primordial cell standpoint, because it does not require the evolution, the development, of a dedicated machinery. But we think also that this mechanism has likely imposed evolutionary constraint on bacteria in terms of the regulation of the cell width and also on the crowding, since those two properties are important for the mechanism that we propose. And so I just want to finish with acknowledging, so I acknowledge the people that did the work and they're highlighted here in black. I also want to thank my entire lab for being a fantastic group to work with. Particularly grateful for funding, in this particular case from Shishimai. I am grateful for your that you came today, and I look forward to discussing this. I'm happy to um, to try to answer any of your questions. Thank you.
Thank you, Christine, for a wonderful talk. And usually we have questions, we put them in the chat, or we just raise our hand. And there is already one question from Kui. You want to unmute yourself and ask your question?
Okay. Yeah, I just have a very quick question that this reminded me about the eukaryotic cell. Do you think that during the mitosis that the chromosome condensation could see that some proteins interaction make that at the nuclear plasma are as a poor solvent for the DNA? And when they are separate and they become a good solvent for them. That following that your talk, I think that this is what I'm guessing.
So I'm not sure. Please correct me if I misunderstood your question. But this relates to a previous finding where we proposed that uh, that the cytoplasm behaves as a poor solvent. And one component for this is the polysome. You're absolutely right, Huey. Which is that I didn't show this, but this was actually first proposed by Jim Weissar, I believe, and probably from others, is that the polysome themselves, because they're excluded from the nucleoid, then it's going to create, like an asymmetry, some kind of osmotic pressure that where it's going to compact the nucleoid. And so so they contribute to this, apparent poor solvent quality of the cytoplasm. So not only the polysome are going to be important for nucleoid segregation, we believe, but also is going to be important for the compaction. So that to me, it's really beautiful because everything goes back to the DNA, right? So the DNA basically is responsible, contribute to the making of those polysomes, and this in turn is going to affect, its compaction and also its segregation.
Okay, thank you very much. Yeah.
Thanks.
Yuping.
Hi, Christine. That was a really beautiful talk. I have two questions. My first question is that what happens in cells that are brown? I think you showed an example of bacillus that is sort of the cell wall-less. But what if there is a cell wall? How do the segregation happen? And second. My second question.
Can you elaborate about this? I didn't fully understand could you repeat that first question?
Right. Would cells that are spherical also?
Yes, I can answer that. Yes.
Right. My second question is what determines the characteristic length of these chromosome blobs? And that's my two questions.
Okay,
Thank
so
you.
the first question, which is a great one, what about cell shape, right? Not all bacteria have this rod shape of that we see in E. coli or in Bacillus subtilis. Well, for spherical bacteria, what is really interesting is that spherical bacteria, and if to my knowledge, and somebody can correct me if I'm wrong, they're actually quite small. So they are small, if you had, it would not work if basically the sphere is too big relative to the DNA, right? But those spherical cells, when they grow, you create a long axis, right? Because the way you grow from both ends, right? And now you actually, it's the same as rod shape, right? So you have a long axis. So you do create the same effect. What it cannot happen, and also the shape, so the width of bacteria like E. coli and Bacillus subtilis actually changes with the growth rate. But the size of the nucleoid also changes because you have, multiple rounds of DNA replication. And we and others have shown that you have a scaling between nucleoid size and cell size. So you avoid that problem. It's only in those
I see.
When you remove the cell wall, the cell wall becomes so big, so now you have a problem. And so I think that there is an evolutionary constraint on cell size, particularly cell width. Great question. Oh, and the second one was, oh no, gosh, you have to remind me.
What sets the characteristics length of the DNA blobs?
Oh, what is the characteristic nucleon length before we see that splitting? You want a number? Is that what you ask?
No, I was just wondering what factors would weigh in to determine how big those blobs are.
That is a
I
great
mean,
question.
they could be potentially smaller or bigger, but
I think
why
I
is it
think
the way?
I think that's a great question. I think this is going to be impacted by the rate of by the Poisson diffusion. And I think this is an excellent question to look with our model, because I think that whether they diffuse faster or slower, I think this is going to that's going to affect when we're going to have that phase separation occur. But this is actually a guess, and I think it's an excellent question for to test on the modeling. And then maybe we can go back to experiments. Yeah, great suggestion. Thank you.
Super cool. Thank you.
Gabriel, I don't know. Yeah.
I was first.
Okay.
So I have two questions, but I will answer. I will ask only one and then the other one later. So Bamshi can go then after me. So my question is a little bit, so from your model and the images of where the polysomes are, from the images, it looks like in the center, it gets brighter than what you have at the pole.
That's right.
And it gets much brighter. And even maybe does it get dimmer at the poles? And in the model, it stays the same on the pole and you get the same density. And so I was wondering also, you said it's harder to escape from there, but it looks almost like things are even drawn to there. So do you think
Yeah.
have you tried to add like some interaction or affinity between polysomes or something to the model?
It's a really good point. And I think that there is potentially contributing factor. The first one is simply the fact that are you still there? Yes, okay, because you
Yeah.
froze for a second. So
Sorry.
that the cell poles are actually smaller, right? The volume is smaller. So when Alex correct for the cell volume, right? Because you have like a bigger volume there than there,
Okay.
then it's going to be higher, like the concentration at the poles is going to appear higher than it does in our image,
Okay.
in our camera graph. The second one, you are right. So what we are saying is that you could have uniform polysome distribution and it still would work. But in reality, they might be localized polysome production. And there is two ways that this could happen. One is that the way that the nucleoid is organized in E. coli is that the original replication is actually in the middle of the cells. And so this is the first region that's going to be duplicated. So right there, you're going to have twofold increase, in DNA, and therefore potentially in polysome production.
Yeah.
Second, in this independently of the gene dosage effect, we know that highly expressed genes are concentrated close to the original replication. So again, this is only going to reinforce the process by creating more polysome production in this location. But even if we have uniform polysome production, the model tells us that it could still work. But I do think that in reality, there is an increased localization. There is an increased production of polysome there.
Thank you. Bamshi, did you still have a question?
Yeah.
Okay.
Thank you. It was a very nice talk, Christine. I was wondering, is there any critical amount of the polysome that is good for segregation, and below that threshold you don't have the segregation at the center?
Absolutely. So and in fact, at very slow growth rate, the accumulation is very low, and then it's going to and the segregation is very slow and very late, of course. From the model, yes, it has to reach a critical polysome accumulation in the middle to have this phase separation to occurs. So the model says yes, and I think that probably that's a good thing, right? Because if, for example, you lose nutrient, you still need to have gene expression, but your polysome production is probably too low to result in this splitting. You see what I mean? So
Yeah.
yes, definitely, you need a certain concentration of polysome to have this phenomenon occurring.
At that stage, if you were to inhibit like translation, like by cyclohexamide or something, is that good enough to forward towards the division, or the cells would arrest in a static mode at that stage?
So the model says yes, it would still work, but not as well for sure, because the mRNA are smaller than the polysomes, so you have a lesser effect. Experimentally, we haven't been able to do this experiment. I mean, the way that we know this could be trivial. We could just affect initiation of translation, right? And we can use drug as you propose. But the problem with this is that it also results in the upregulation of ribosomal RNA. So we have a confounding factor right now, and we haven't found a way to test it. So from the modeling standpoint, you say it should still work, not as well. From an experimental standpoint, we haven't tested it rigorously.
Okay, thank you.
Great question.
And we have a question by Hugh.
I think he's there might they say they can speak.
Oh, sorry. Yeah. Thank you for the talk. Could you please explain why polysome phase separate accumulate in the middle but not at the cell periphery?
Yeah.
And why won't it happen in mammalian cells?
Okay, let me answer the first question. So the reason is this is why you need to reach a certain nucleon length. It's simply because the probability of escaping from the nucleoid is lower if the polysome are formed in the middle than if they are formed, like on the edges, right? And so it's going to establish a gradient that is going to be the highest in the middle of the nucleoid, hence the accumulation there. But going back to Gabriel, the discussion that we were having with Gabriel, there might be a reinforcement of this mechanism due to an increased gene expression occurring in the middle of the nucleoid. That's also a possibility. Yeah. And what was mammalian cells?
Why not in mammalian cells?
Well, in mammalian cells, as I understand it, transcription and translation are mostly, separated, right? So, but I do think that the mRNA and the DNA could have, a repulsion sterically, certainly, and even potentially non-sterically. There is Dr. Joyeux that basically makes the argument that there can be a phase separation between RNA and DNA in the nucleus. So I think that something similar could also happen, but with the difference, of course, that in bacteria, and this is the important point, there was no nuclear membrane. So the ribosome can go diffuse through the nucleoid, load up on the mRNA, and now you have that steric clash with the polysome because they are formed specifically. They can already be formed in the nucleoid.
Good. Then we have Tomoko. Yeah.
Oh yes. Thank you so much for your great talk. I have two questions. So first thing is I was naively wondering, do DNAs get entangled during the division, and then if so, how is it overcome?
Yes, and in fact, in my last slide, and I forgot to mention the point, the mechanism that we discuss here is only contributes, right? I do think that there is other factors, and those other factors I think are going to be particularly important for disentangling the DNA. So, for example, in our model, the DNA is considered as a solution of DNA fragment, and that can correct me if I'm wrong, which means that there is no disentanglement. So we have a phase separation, but it's because it's considered as a solution. Now, in reality, this is a polymer, so it need to be disentangled. And I do think that there, DNA extrusion loop, with the condensate is going to be very important. It's also going to be important for breaking the asymmetry. And I think the PyBS system is also important in separating the origin. And that provides, you break the asymmetry so that now when you have the split, well, you split the two things apart, right? So I think that there is other factors such as DNA replication. DNA replication is also going to help disentangle DNA loop extrusion, and I think are going to contribute to the disentanglement. In our case, our mechanism does not help there, in my view.
I see. Thank you so much. The second one is that does the number of ribosomes on RNA in the polysome matter or make changes in terms of the separation ability of nucleotides?
I think that's a brilliant question. I agree. I think it has to, right? Because the more ribosome you are going to have, and the bigger it's going to be, and the more you're going to have this steric repulsion effect. So right now in the model, we have only one diffusion coefficient, which is an average, right? So now you just consider the average case for the polysome. But you're right. I think that polysome of different sizes are going to have different effect. And in fact, I show you, like, we think that the mesh size of the nucleoid is heterogeneous, right? What we did was Jun Liu at Yale, we did cryotomography, and we look at the localization of ribosomes. And by looking at how they are arranged, we could even identify the polysome, specifically the polysome. And you can see that some of them accumulate in some places within the nucleus more than others. So is this dictated by the size of those polysomes? It's entirely possible. I think that it should have an effect, yes. And I think this is through modeling, it could be examined, right? We could just look at a range. And in fact, I think that she actually look at this. He changed the range and he saw, that of course you can have different effect depending on the size. The bigger the size, the stronger the effect.
I see. Yeah, that's super interesting. I thought it could be interesting to see if this effect comes from the polysome as a whole or just the ribosomes could. Yeah, I was curious if the fact that the multiple ribosomes are connected with mRNA is important, or is the bulkiness of the ribosome is important for this separation ability?
It's a very good question. Right now, we're only considering steric effect, right? But there might be also non-steric effect, from the mRNA, from the ribosome. And going back to the previous question, we have not disentangled those effects. But what we're arguing is that even just sterically, it can work, even if we consider the steric effect.
I see. Thank you so much.
Gabrielle, you muted.
It's not helping to moderate when I'm unmuted. So June has another question. That's all I wanted to say. June?
Oh, okay. Thank you. Can you hear me?
Yes.
Okay. Thank you. Great talk. I have a short question because I'm curious about, like, in the fast growing E. coli cells, if they have multiple replicated genome in a cell, and based on your polysome exclusion model for the DNA segregation, I think it's basically, I think it may be mainly rely on the amount of the DNA or the mass of the DNA. But in that case, if there are multiple replication happening, how can you make sure, like, the DNA will not only be segregated by their mass or amount, but by the biological meaningful genome, more wholesome, but not more like cut the DNA just directly by the symmetrical division controlled by the DNA mass. Yeah, I'm not sure if I ask us a clear question. Yeah.
Yeah, it's really on me. I'm not sure that I fully understand, but you're right? So that when the bacteria grow really fast, they can be born with two nucleoids. So nuclear segregation can even happen in the preceding division, which is what, does this make sense? Do you know what I mean? So in bacteria, right, you can have multiple fork of replication ongoing. And the reason is because, just to give a little bit of background, I mean, bacteria can do this amazing thing where they can actually divide faster than they can replicate their DNA. And the way that they do this is because they can initiate DNA replication in the previous generation, or sometimes 2 generations before. Okay, and then they finish it in the later. So the start of DNA replication and the end of DNA replication occurs at the different generation, which is absolutely remarkable. And they deal with this also by segregating the nucleoid earlier, right? So you could have nucleoid segregation occurring, in the preceding generation. And that is not a problem, because as soon as you have the 2 nucleoid being formed, right? And before even the cells divide, then now you have the polysome accumulating. And we see this at the 1/4 to 3/4 position, because it's the middle of those new nucleoid that was just formed. And so then the cells divide, and now the cells divide with already 2 nucleoids, right? But that's fine, right? Because nuclear segregation, DNA replication, nuclear segregation is completely independent of the cell division cycle. And so, they can handle it this way. Now, what question were you maybe I'm sure I didn't answer your question.
Okay. Yeah. Actually, I mean, basically, I'm just to imagine if there is a case if the polysome can like push the not replicated, can push some replicating genome and cut it to separate it, to separate the genome directly based on the DNA amount, but not by like what we want is the replicating DNA should still stay on the replicating genome, things like that. I'm okay. I think
Yeah, what we're arguing is that you have DNA replication, right? And you can have some disentanglement, but that alone is not going to push the two nucleotides away. You need something to push them away from each other. And this is where we think that the polysome are going to contribute, right? Yeah, otherwise you can't, they could stay next to each other, right? But how are they going to move away? And they have to move away, right? So that you can divide and then you can continue the process.
Okay, thank you so much.
Yeah.
Thank you.
I wanted to ask another question, and that is, so you mentioned the lateral attachments of that through basically translation, and that also sounds like a great way to contribute somehow to some degree, right? And so I was wondering, in your experiment where you prevented transcription, then I was amazed how rapidly that resulted also in like complete stop of growth, basically, right?
Mm-hmm.
So then you also lost this contribution. So can you somehow disentangle this? Yes,
this is a great point. So originally, like the first model that was published by Jacob is that it was simply growth, right? That the nucleoid is attached, right, through this translation process, which is transcription, translation, insertion of nascent, transmembrane protein to the wall, right, to the cell envelope, and then as the cell is growing, then the nucleoid are moving. It's beautiful because now you also have this interesting intrinsic coupling. However, it could not work, and this has been debunked more than once for two reasons. First of all, the model assumes that you would have localized incorporation of growth, right, in between to separate them. And we know that it's everywhere, right? So that doesn't help. The second thing is that the insertion is to the membrane. The membrane is very fluid. Then again, that does not help because it would have to be connected to the cell wall. Yeah. But then again, the cell wall does not grow, in this polarized fashion. And then the third point is that chromosome segregation, especially in fast proliferating bacteria, chromosome segregation is faster than growth. So it cannot be that. However, Alex's data show very nicely that at the very end, there is actually insertion of peptidoglycan material for the division, right, of the cell wall for the division. And now we think that there is a contribution for growth because you have this localized growth.
Okay.
So we think that they participate to the last step, but it cannot explain the whole process.
Thanks. And
but that would have been very neat if it happened. Yeah, it was proposed back in the 60s, I think. Yes.
Okay, yeah. I was like, it's an obvious one in a way.
Yeah, it is super cool.
Okay, okay. But one thing that I wanted to ask you before also, so the polysomes, right? So can you exclude that there is some kind of affinity also between polysomes, that they, there's not only like repulsion, but there's also like attraction?
Yeah, I think that this is really interesting, that there can be all those non-steric effects. I believe, but that's more like based on faith and desire, that there is actually non-steric interaction, but I don't know. Yes, because we do see this, right? We see this kind of like phase separation like phenomena, right? Where they get together, and in fact, I could show you like other cases where we make the cells, kind of fat and elongated. And now the polysome can diffuse each other, and then they basically accumulate to bigger patches. So it looks like it. Yes, I agree.
They will still come together. Okay,
yes.
But I mean, I was just wondering if you in the model you could just like maybe add this and then just see what would happen. I mean, that doesn't mean there's
in the model. This is pure phase separation, so it would definitely happen in the model. So
Okay.
this is a spinodal. So it is a phase separation in the model, definitely, but the model doesn't have the polymer, right? It's really a solution of DNA. Yeah.
Yeah, but in the model, it will basically just happen because of steric exclusion. You will get phase separation, but it could be enhanced if you had like attractive interactions, I guess, of the polysomes or something like that.
So in the model that Ned did, it was a 1D model, so we cannot have communication.
Okay.
But I do believe that in Jan's model, and I see he has a question, it is 2D, and then you can look at those kind of effect.
Okay.
Yeah.
Good. Thank you. Yeah. Sorry, Jan, I didn't know whether you had a question.
No, I was
But
also saying that we have 3D model using
3D
lamps.
model, not just 2D model, 3D model, yes.
So we still see there, but now we're looking at more details, and I guess there are some things which is different in 1D, which we haven't published yet, but I guess the published one is kind of conformative of 1D model. And so there is only repulsive interactions indeed between ribosomes. And I guess, like Christine said, that this diffusion of polysomes is rather slow in the cell, and that allows them to accumulate in the middle, even though they could diffuse around the nucleoids, around the periphery, around the cell wall. But that seems to be quite slow process, perhaps also because of these transition linkages. So, but the other thing is that there are our work shows that there is kind of many different mechanisms, and this is one of them. So that there is also like FTSK, which is kind of translocase, and that is activated in very late stage. And if there is a DNA trapped, it kind of can pump like megabases of DNA, and the constriction itself, the process can also help in it. So I guess biology relies on multiple kind of concurrent processes. This is one of them.
Cool.
But great talk, Christine. I really enjoyed
it. Thanks, Jan.
All right. So thank you all who stuck around all the way.
Yeah, thank you
to the
so
end.
much.
If we have no more questions, let's give Christine another big applause.
Thank you. Thank you all for coming, and thank you for the great question. Great suggestion.
Thank you. And then I hope to see you soon somewhere.
I'm sure in some conferences. Thank you all.
Good. All right.
Take care.
Thanks.
Bye-bye.
Have a good week. Ciao, ciao.
You too. Bye.
Bye.